My name is Ruchira Mirji, and I am a biological sciences student concentrating on genetics, cell, and developmental biology. Since my concentration is on genetics, I wanted to focus on the research regarding finding a cure for Alzheimer's. Throughout my presentation, I will be discussing genetic engineering and, most importantly, reducing TAU protein levels for Alzheimer's disease. The news article in which I found the scientific journal and the primary source was from News Medical Life Sciences. I decided to choose this topic mainly because I am currently living with my grandmother, who currently has this condition, unfortunately. I haven't been able to study too much and research regarding this, but this class and assignment gave me a chance to really dive deep into understanding what is being done to prevent such a horrific disease. Before diving into the news and scientific journals, let us go over what exactly Alzheimer's disease is. Alzheimer's is a form of dementia and is a very progressive disease. It is heritable, meaning that it is only passed down by family members. It is essentially a progressive disease that affects the individual's memory and causes severe memory loss in the last stage. Unfortunately, there's no real cure for this disease. However, there are still ongoing treatments to help prevent and slow down the process of this progression. So, what causes Alzheimer's disease? There is a protein called TAUA protein that is generated in the brain. According to the news articles, the Massachusetts General Hospital, as well as the Sangamo Therapeutics Inc., have taken part in leading this investigation or experiment. This protein, which is coded, essentially causes entanglement and catalyzes a start to developing the disease in the brain. Once the disease progresses due to the protein production, it causes synaptic loss, neuronal loss, as well as cognitive decline. So, what treatment is being used to help the betterment of this disease and the patients currently suffering? Researchers have developed a new technology called the zinc finger protein transcription factors. This is a gene regulation technology that essentially affects gene sequencing. Taking a look at the primary literature article, the zinc finger protein transcription factor is actually a form of recombinant adeno-associated virus, which helps transduce cells in adult central nervous systems. This means that it transfers genetic material from one host to another to incorporate the genetic material. The goal is to use the certain technology and silence the gene expression that codes for the TAUA protein. With this, researchers have brought in mice that have been injected with disease and the TAUA protein. The zinc finger protein transcription factor is then injected into the mice directly into the hippocampus to view better results. As we can clearly see in this diagram, there are several diagrams within which all help us show the steps which is being taken to eliminate the TAUA protein. In image A, the zinc finger protein transcription factor has recognized the DNA sequence or gene expression which codes for the TAUA protein, and in section B starts to bind to the sequence. In this case, the gray bar indicates the location of the TAUA MAPT gene, and the blue arrow represents the direction in which the ZFPTF will be coding in. Section C and D shows the ZFPTF has been coded into the sequence along with the help of the promoter. And image E shows us to view the results of technology applied. As a result of this experiment, the protein levels were reduced significantly from 50% to 80% over the span of 11 months in the experimental design. This showed reversed effects of Alzheimer's disease. Additionally, there were no histopathological changes, molecular alterations, and further neurological damages due to this protein technology. Although they have received such great outcomes and accomplished results, this is yet to be tested on humans as it is only experimented upon animals such as mice. While reviewing both sources, the news, medical, life sciences, as well as the primary literature, there can be a there can be seen a significant difference in amount of information and data which have been provided by each. The news article only provides a brief summary and outlines the basic and general idea of the lab, whereas in the primary literature, there are very detailed information regarding the outcomes and results. The primary literature supports these results with diagrams and further data analysis for the audience to understand. Although the news article does not hold valuable information, it lacks many details in understanding the process and procedure of how the new zinc finger protein technology factor works in the hippocampus and what kind of protein this is.